So we're really lucky this morning to have Dr. Michael Rosenblum as our speaker at the clinical trial seminar. And he's going to talk to us about statistical challenges in the design and analysis of randomized trials for learning which populations benefit from different treatments, which I think I get asked that question all the time, not the statistical part, but how do we put evidence-based medicine together with personalized medicine? So you're going to answer it for us, right? And
say a little bit.
Dr. Rosenblum is in the Department of Biostatistics here. He is an expert in adaptive design, so many of you may know him from that particular topic. He did his PhD at MIT in applied mathematics and probability, and then went on to do a postdoc at UC Berkeley in biostatistics. He works in HIV, AIDS, and some of you may know him from that work here. As I already said, his interests are in adaptive design, robustness to model misspecifications, and causal inference. So with that, I'll hand it over to Michael. Thank you.
Great. Thank you very much. It's an honor to be here. Thank you all for coming and for getting up early to come to the talk. I'll be discussing some work I've done and with many colleagues on design and analysis of randomized trials, where the goal is to learn not only whether it's better to give treatment A to everyone or give treatment B to everyone, but also to try to learn who in a population is benefiting from different treatments. The outline of the talk will be, first I'll just give an overview of adaptive designs and some of the statistical challenges that are out there. There are many really exciting open problems, so I want to try to generate some excitement and get people interested to work on some of these problems. I'll talk about a recent collaboration with the FDA that completed in September and some of the results of that collaboration. And then I'll focus in on one particular result, which are new group sequential adaptive enrichment designs, and I'll say what that is exactly and give some examples. So that's something that I and colleagues developed along with open source software to allow people to plan clinical trials, or essentially compare different types of designs tailored to their specific scientific question and logistical constraints to see if you can get some advantages out of adaptive designs. So that's, I think, one of the big open questions is when, for a particular researcher with a particular scientific question, they'd like to know, they might want to know what's a good design to answer my question. Currently, it's pretty hard to figure that out. There are just many, many designs out there that you could kind of try this one or try that one. It's a trial and error approach. But I have some software, and I'm working on expanding it to allow people to do a head-to-head comparison very quickly of many types of designs to try to optimize to their problem. And then I'll talk about future research directions. So first, I'll give the overview of just general adaptive designs. So pharmaceutical companies are one group that's particularly interested in adaptive designs. And this is a clipping from a newsletter called the Clinical Trials Advisor from a few years ago. And the headline is Adaptive Trial Designs Save Merck Millions. And the first line of the article is, an adaptive clinical trial conducted by Merck saved the company $70.8 million, compared with what a hypothetical traditionally designed study would have cost. So it's not actually true. It's more accurately, it's that a hypothetical adaptive clinical trial. So they essentially look back on a completed trial. So I contacted the author of this article to try to find out more details, and they said, well, it's a hypothetical, and it actually tells in the article as well that it's a hypothetical sort of retrospective analysis looking at the completed trial and what would have happened if an adaptive design had been used. And I'm not particularly criticizing that approach. I think can be a good approach to try to look at the pros and cons of different trial designs. And can anyone guess where that 70.8 million came from? How could you save 70.8 million? It's a pretty big chunk of money. Or how are they coming up? What was it that allowed them to save a bunch of money like that, by at least hypothetical money? Yes.
Reduce number of participants?
So it is reducing the number of participants. That's right. And how could that lead to $70 million? Exactly. So they saved some money on enrolling fewer patients. And you could also argue that there's some ethical benefits to having fewer patients in the trial. But the big benefit came from they claimed they could reduce the expected duration by 2 months of the trial. And that would give their company 2 more months on patent of their drug. And for a blockbuster drug, they computed that that's 2 months extra time on patent gives you 70.8 million. So it was really a time reduction that saved them. So at least in this case, that was what gave it. Jay?
Yeah, I think it's only fair to say, though, that 70.8 doesn't take into account the increased operational costs that an adaptive design would have had. I mean, I'm in favor of all this, but when people throw out numbers like this, they often they don't do the subtracting.
That's true. I mean, this is all.
Yeah.
This is nothing to do with me particularly. But yeah, no, it's always, I think, going to be a trade-off. Yeah. It's not. I'm certainly not saying that adaptive designs are the answer that will solve all your problems. Sometimes it would be a terrible idea to do an adaptive design. But what I propose is to develop new methods and construct open source software. I'll give some examples of that to allow people to compare head-to-head standard designs to adaptive designs and see, well, what are the potential benefits and potential limitations of different designs. And so when I presented this a few years ago at it was a workshop on developing new antiretroviral drugs for HIV treatment, and I said that I apologize to anyone here from Merck. If there's anyone here, I'm not trying to say anything bad about Merck. And then the next speaker was from Merck, which I would have known if I had looked at the list of speakers. I mean, it was very clearly written there. And then afterwards, I went up and apologized to that speaker from Merck, and he said, "Oh well, nope, it's not a problem." And also said that actually at Merck, if you don't include an adaptive component in your design, then you have to say why not. You have to give some rationale for why you didn't do it. So, and that's just anecdotal. That's just one person telling me this, but I mean, I think that's a little bit evidence of adaptive designs being a little overhyped. They're certainly not the solution to every problem. You shouldn't just jump to it. But there is potential, I think, to have substantial benefits in some cases. And part of the problem is to figure out, are you in that case where you have a lot to benefit or not? So the potential benefits of adaptive designs are you might be able to get more power to confirm effective drugs. What I focus a lot on is you might be able to better determine which subpopulations benefit from a particular treatment by using these designs. And there's potential to reduce cost, duration, number of subjects in the trial. There are certain statistical properties. This is not the only things that you would want to have, but at least a few key things. And these are highlighted by the FDA in their draft guidance on adaptive designs, which I'll talk about in a minute. One key thing is controlling the probability of a false positive, the type 1 error control. So you need to have that. But there's also issues of controlling bias and also ensuring that you can construct confidence intervals for whatever quantity you're estimating at the end of the trial that have correct coverage probability. So even doing a standard group sequential design, you induce some bias in your estimate at the end of the trial. So the worry is that maybe things could get worse in adaptive designs. There are some results to show in some cases it will not happen, but those are some issues. And I'll be talking about the setting of group sequential randomized trial designs where patients are enrolled over time, and at interim analyses that are pre-planned, there's an option to change some part of the sampling in response to the data that's accrued. And in all of what I talk about, all of these designs, the adaptation is pre-planned. There's some decision rule that's been written up in the study protocol, and the focus is on adequate and well-controlled trials, so essentially phase 3, phase 2B, or phase 3 trials. And there are many types of things you could adapt in a design. You could adapt the sample size, you could adapt the randomization probabilities, but what I focus on is adapting who is enrolled in the trial. So I focus on the patient enrollment. And the FDA has a draft. They have multiple draft guidances on adaptive designs. This is from the draft guidance for drugs and biologics. And I highly recommend this document to anyone. It's very clearly written. There's a 2010 and a 2012 FDA draft guidances. They're both very well written and explain clearly what are the things that are well accepted. They're called well understood types of designs, and which are the less well understood designs. And the ones I'm work on are the less well understood. So it essentially lays out a bunch of research problems that if you could figure out some of the properties, improve some properties of these less well understood designs, you'd be making a contribution that could be valuable. And so a quote from the FDA draft guidance, the 2010 guidance, is that for, in particular, types of designs I work on called enrichment designs, that's where you can change the population enrolled. You might restrict it to a certain group if there's some early evidence that the treatment only benefits that group. That these enrichment designs are less well understood, pose challenges in avoiding introduction of bias, and generally call for statistical adjustment to avoid increasing the type 1 error rate. So you have to be very careful when you have these designs. And so a lot of the methods I work on are to solve these problems. I'll talk briefly, just a few minutes, about a wonderful collaboration that I was part of over the past few years. It recently ended in September when the grant finished. And it's called PACES, the Partnership in Applied Comparative Effectiveness Science. It was a partnership between Johns Hopkins and the FDA. And the PI was Jody Siegel here at Hopkins. And there were 4 teams in this in the PACES project, and I led one of the teams. And so I'll just talk about that chunk of it. But the whole project it was wonderful for me to be a part of it. That our team focused on randomized trial design and analysis for estimating treatment effects in subpopulations, essentially what I'm talking about today. And there are many people involved. It's myself and Scott Zieger, and then 3 students, Huitong Qiu, Ching Tsai, and Emily Huang, working with me on different parts of this. And at the FDA, there were statisticians, some brilliant statisticians, Estelle Russick-Cohen, Bob O'Neill, Dan Rubin, Greg Soon, and Sujane Wang, we were having monthly phone calls. So I could talk to them about would it be useful if I solved this problem? Or can you tell me some of the problems that would be most useful to solve from the FDA perspective? And they were great at giving advice and ideas. And there's Sam Ogunbo at a company, GDIT. He was also instrumental. I want to give him special thanks because he went to the FDA, and FDA essentially let him do some cleaning of their data in HIV trial data, which was then given to me. But he and Greg Soon and their group did amazing work on this. And the specific aim for our team was to develop statistical methods and software to determine the best trial designs and analyses for generating evidence about treatment effectiveness in different subpopulations. And the type of populations, well, I'll tell in a minute a little more specifics, but just the summary of the outputs of this project are 6 papers published or under review. They're mostly just under review, but some have been published. And the papers, this is essentially to give an overview of the types of things that I work on. So if there's anything here that you have questions on, feel free to come up and talk to me afterwards. I'll focus on one particular paper, the results in that. And everything else is all available on my webpage for anyone who's interested. But there are results on new adaptive enrichment designs, how to construct confidence intervals for adaptive enrichment design. So that was generally an open problem. If you have this selection, you're selecting which population to continue enrollment of. You might induce some selection bias, which can screw up the confidence interval coverage. Sometimes it can mess it up substantially unless you do some kind of fix. And then we try to find the fix that minimally expands the width of the confidence interval. New adaptive group sequential designs that can combine advantages of standard group sequential designs with adaptive enrichment. That's what I'll talk about today. There are new optimal testing procedures for multiple populations. So even in a standard trial, there's still questions about what is the best testing procedure to use at the end of the trial to analyze the data to learn about which populations benefit while still controlling the type 1 error. So we give new optimal procedures for that. And then another output is a user-friendly software tool that implements some of these new methods. And the idea is to use this tool to plan a trial or to essentially compare trials for meeting specific goals. And then if a trial is a new design is advantageous, then it allows you to plan it out a little bit. And so some of the results, there's a paper on optimizing trial designs to learn which populations benefit from treatment. This is an example of a new class of adaptive enrichment designs. These are two-stage designs, but they're quite flexible that allow you to decide to switch enrollment to just a certain population. There's a paper on how to construct confidence intervals for the population that was selected to continue in a trial that has correct coverage. And then we show that you only need to expand the amount you need. Any guesses, actually? Well, I haven't given enough details to ask this question, but how much you'd have to expand the confidence interval, the standard naive confidence interval, in order to guarantee correct coverage in an adaptive design. It turns out for these designs we look at, you only have to expand it about 5%. So it's not so terrible. There's new optimal multiple testing procedures for learning about treatment effects in the combined overall population and two subpopulations in randomized trials. And it turns out that even though there are just 3 hypotheses you're interested in, the problem is extremely hard computationally to solve, and essentially because we want strong control of the type 1 error rate, meaning that no matter what the true data-generating distributions are, no matter whether one population benefits, one has no effect, maybe they both benefit, maybe neither benefit, maybe one is harmed, one is helped. You don't want to make any assumptions up front. And if you want to control the type 1 error and optimize your power under a few alternatives, that's an unsolved problem, how to do that. So we give a solution to that using some methods in sparse linear programming and find optimal designs. And we show that actually there's some nice trade-offs that can actually boost it. You can has a practical side in it. You could boost up your power for detecting subpopulation effects while only a little bit reducing power for the overall detecting effects in the overall population. There's a pretty favorable trade-off. What I'll talk about today is adaptive group sequential designs that balance the benefits and risks of expanding inclusion criteria in a trial. I'll just skip over these last two, but there are some, I'll at least mention this one with some collaborators that Brandon Luber, a student here, and Antoine Chambaz, a collaborator, we have a paper on trial designs that simultaneously optimize the population that's enrolled and the randomization probabilities. What I'll be focused on the rest of the talk is just changing who's enrolled. It's always 50-50 randomization. But here, there's multiple adaptive components. And we actually show that in terms of power, you don't get much advantage. But in terms of the number of patients exposed to the inferior treatment, you can get a nice advantage. So that's something that I think has not been the focus of adaptive designs necessarily, is minimizing patient exposure to a harmful or the inferior treatment. But actually, sometimes this combination can help with that. So a common thread in the work that I do is the subpopulations have to be predefined. So as well as the adaptation rule. Everything has to be set up front. It's not the case where you can sort of be in the middle of the trial and do whatever you want. It has to all be preset. There are people who work on trying to learn the subpopulation as you go. That's a harder problem. It's an important problem, but that's harder. So I'm always looking at the case where you have prior data or some biological knowledge that allows you to conjecture that the treatment effect might differ in certain populations. And I've worked on actual trial designs where that seems pretty reasonable. The goal is to test hypotheses for the subpopulation treatment effects, test the null hypothesis of no effect for some subpopulations, and then for the overall population. And also you'd like to estimate these effects. Always I ensure that you have strong control of the family-wise type 1 error rate, control the false positive rate, and that you can prove that. I won't get into some details, but there are some types of designs that are more ambitious than what I do, but where you can't prove that you have strong control of the type 1 error rate, but it's a trade-off. But in all the designs I do, at least, you can always prove you have the strong control. And some of the potential advantages of these designs are, at least in the cases that we've looked at, which are based on resampling from real data sets and looking at the performance of different designs, that you can potentially get more power, smaller expected sample size, or more participants assigned to the better treatment arm. And generally the goal, and you could sometimes get this, is generating stronger evidence about who benefits from a treatment. So that's really the end. That's the thing I'm most excited about. Is you can try to steer the trial to learn a little bit better than a standard trial who is benefiting. So before getting into this, I'll talk about one of the papers and one of the new types of designs and statistical software implementing it, allowing you to plan trials using this method. What's wrong with a standard randomized trial? I mean, that's the simplest. So if that suits your needs best, you should just go with that, for sure. So just as a special case, as just a thought experiment, think about the case where the overall population you're interested in is sliced down the middle, and you have two subpopulations of equal size. And if the treatment effect is equal in both groups, then typically the standard trial design is going to be the best design. But if the treatment only helps one of those groups, say the treatment is ineffective for one population, but for the other population it helps. And there are a bunch of examples where that's thought to be the case. There's a particular group benefiting from a particular treatment. And I'll give one example later on, but that's not the only one. So in that case, where one group benefits and the other doesn't, and here I'll assume equal variances in the two groups, then a standard randomized trial can have quite low power. Basically, if the treatment just benefits one subpopulation. So if you powered the trial at 80%, so it has 80% power to detect a benefit in both populations, so you're looking at just the combined population, and you power it so that if the treatment effect overall is a certain clinically meaningful level, that you have 80%, so you set your sample size that way, but in fact it turns out that only one of the populations, half the population here, benefits. The other there's no effect. Now your power dropped down to 35%. And you're not only that, but you're not necessarily going to learn that the treatment only benefits this one group. You might reject the null hypothesis for the combined population, and now say everyone can get this drug, but you might not even learn who benefits. And the issue is that in a standard randomized trial, you're essentially locked into either keep enrolling everyone or just shut down the whole trial. There's not typically an option built in to just continue with one population. So that's the issue that we're trying to address. And a particular example that I worked on, and this is a collaboration with Dan Hanley, here, and it's in the context of a stroke trial, and this is actually a trial that's about to start. Not what I'm going to explain, this adaptive component, but this is what we looked at. But this is in planning this phase 3 trial, the goal is to evaluate a new surgical treatment for intracerebral hemorrhage, which is this quite deadly form of stroke, has 40% mortality rate, and at least. And Dr. Hanley and others have proposed a new surgical treatment for this form of stroke, and it's called the MISTY treatment, which stands for minimally invasive surgery plus RTPA for intracerebral hemorrhage evacuation. So I'll do a terrible job explaining what is happening, but from my layperson perspective, they do some imaging, so a CAT scan, and then they drill a hole in your head, and then they suck out. They put in TPA and try to evacuate, to break up the clot and get the clot, and then try to aspirate out the clot. And the phase 2 data is, you can never make any strong conclusions from phase 2 data, but it was impressive. And so we were planning the phase 3 trial, and 2 subpopulations were identified of special interest. And one is, so this is all focusing on intracerebral hemorrhage, but and all the patients in the phase 2 trial essentially were people who had just had this problem and did not have an intraventricular hemorrhage, different type of hemorrhage, either didn't have that or had a relatively small intraventricular hemorrhage. So those are called the small IVH patients. But it was thought, and that was who we had good data on. But it was thought that also people who have this essentially secondary, a large intraventricular hemorrhage in addition to the intracerebral hemorrhage, they also could potentially benefit. There was some medical reasoning that, which is beyond my understanding, that it could be quite helpful there for this population as well. And it would be useful to know, it would be more generalizable result if we could show it actually works for both these groups, or just for the whole population of people with ICH, whether you have an IVH or not. But the risk in including this second population in the trial, phase 3 trial, is that if it doesn't work for them, that could sink the trial. And since there was not that much data, very little data on large IVH, people with large IVH at baseline, at least I felt that was pretty risky to put the success of your trial, to hinge it on a group that you didn't have much data. So it would be nice to build in. The idea was, could we build in some mechanism where if partway through the trial, this large IVH population, if they were not benefiting, if there was just no signal far enough along in the trial, or if there were some unexpected adverse events that were worrisome, that you could just stop that group and then shift to focusing only on the patients with small IVH at baseline. But you still want to control type 1 error, have a valid interpretation at the end. So it's tricky, and you want it all pre-planned. And we make it a little bit flexible in the sense that if there's any adverse event issue, you can always stop enrolling the large IVH for any reason. Still, the statistical integrity of the trial is maintained. That you control type 1 error under any choice there. So I'll give an example of what we came up with I'll tell the goals, the specific goals, and then an optimized trial design to try to meet those goals and compare it to standard designs. But the general idea is that there was much greater uncertainty for large IVH participants. There's just not that much data on these guys yet. But it would be great to include them in the trial because we'd like to know the answer. It would be, and actually they make up their thought that the projection for what proportion of the overall population of people with ICH, what proportion have large IVH, it was thought to be about 2/3. So it's a pretty big chunk of, you would learn a lot more if you could answer this question for people with large IVH, but you don't want to put all your money on that because you don't have much background data. So in planning the phase 3 trial, we'd like to be able to learn, does the new treatment benefit small IVH participants, and also does it benefit the combined population? And first, I'll just give some heuristic ideas about to introduce enrichment designs. This is not exactly what we did, and then I'll tell more details exactly what we did. So you could do a fixed design where you enroll, so say subpopulation 1, the green one, those are the group with small IVH at baseline, small or no IVH, but I would just call them small IVH. Subpopulation 2 are those with large IVH at baseline. So you could do a standard trial, just enroll anyone with ICH comes in, and you could do a standard group sequential design, or just for simplicity, let's just say it's a design that you have a fixed number of patients, and then you do an analysis at the end. That is probably the best thing to do if the treatment has an equal benefit in both groups. But if it only benefits, say, the small IVH population, then the signal will be attenuated quite a bit by subpopulation 2 not having any benefit, so the power will be pretty low. So that's the risk in doing that. And otherwise, an alternative is to do a standard design where you forget about the large IVH population, just enroll small IVH. But that has the disadvantages that you're answering a question for a smaller group. Recruitment will take a lot longer because they only make up about a third of the overall population, so you're going to be waiting a lot more time. So that's the disadvantage there. That you could essentially do this, and then afterwards you could enroll large IVH population. But that's going to take much more time, essentially. So a hybrid approach is to essentially start with enrolling both groups. And if there is a signal that there's not much benefit for population 2, you stop enrolling them and just focus on subpopulation 1. Or if there's adverse events that are higher for subpopulation 2, then you just go to subpopulation 1. So start with this, and you have potential to shift to this, but then you still want to control all the statistical properties. And also you want to avoid stopping a group where there really is a benefit. That would also be a pretty bad mistake. So you can start off the hybrid approach. That this is an example of an adaptive enrichment design. Is you start off recruiting both populations. Say we'll take the simple 2-stage example, but then all of this stuff, actually a contribution that I'll talk about in a little bit, is extending this to essentially seamlessly incorporate group sequential analyses in here. So you can have as many interim analyses, as long as they're pre-planned, as many as you want. And you can leverage a lot of the machinery of standard group sequential designs and combine them with adaptive enrichment. I'll give an example to illustrate that in a minute. But for now, just you could start off, say stage 1 is enrolling both populations, and then you do one analysis in the middle, where if the treatment effect is relatively strong, there's some signal from both populations of a benefit, and there's not a higher adverse event rate for population 2, then you just continue enrolling both. But then there's an option to switch. If the treatment effect there's a signal in one population but just nothing, or negative in the other, you might switch to enrolling just one of them. And you could build in that you also have the option to only enroll subpopulation 2 if things are reversed. I won't talk about that in this example I give, but you could extend things to do that if you wanted that flexibility as well. That's possible. So there's a lot of impressive related work in this area. There's been the initial focus. There's been a lot of work on adapting treatments. So a similar problem to learning which population is when you have more than 2 treatments and you try to adapt. You try to learn which treatment is the best among more than 2 treatments, and so you might give up on certain treatment arms. And it's not the same problem, but it's a related problem as the problem of learning which subpopulations are benefiting, and you might stop certain subpopulations. So there's been a lot of impressive work on that, and I highlighted a few by some of the colleagues that we worked with at FDA. So it was great to have this opportunity to work with some of the people who have come up with these methods and who are at the FDA who deal with the real tough issues of determining which drugs and devices are they going to approve or not. That's much harder than my job. So And then something that's not as much looked at, there's a lot on testing, hypothesis testing, and that's what I'll talk about today. But a little bit, but not as much work has been done on constructing on estimation and on constructing confidence intervals. So I think that's equally important. You want to be able to test if there's an effect, but also you want to be able to estimate it and construct valid confidence intervals at the end. So that it's not that there's been no work. There's certainly been important work on that, but that's a little less worked on. And we have some new methods to answer some to ensure that your confidence intervals have correct coverage in adaptive designs. And so I'll focus. Well, let me just first ask: are there any questions on anything so far? I'm happy to. Yeah. So, yeah, it's a good question. So that generally type 1 error is definitely maintained. And if you, let me just go back for a second. So I have this option, let me see here actually. So if you give up, so the design will allow, that I'll show in a minute, will allow you to essentially give up on large IVH patients for any reason. For example, safety could be an issue. Adverse events could be an issue. And if you do that, then that could hurt your power for those guys. But it won't hurt the power for these guys. So at least you're always going to maintain power, which I set this threshold, I'll show in a minute, for 80% power to detect a certain minimum effect for these small IVH. That is always protected. But adding the flexibility that you could stop larger IVH for any reason, then I can't make any guarantees on the power for that group anyway. Unless you could build in specific rules. Potentially, we could, yeah. But you maintain part of it at least. That's a good question. So I'll talk about for the rest of the talk. This is a paper that's currently under review at Biostatistics, and it's a collaboration with Rick Thompson and Brandon Luber at the Biostatistics Consulting Center, and Dan Hanley, who's the PI of this trial. And the primary outcome, both in the phase 2 and phase 3 trial, is the degree of disability of participants based on the modified Rankin scale. And we call for what we'll talk about, call a score of less or equal to 3 as a successful outcome. It's a disability scale from 0 to 6, where smaller values are better. So that's the primary outcome, and these are all people with ICH, with intracerebral hemorrhage. And the two hypotheses I'll focus on here are the combined population null hypothesis, H0C for combined, which is no mean treatment benefit for the combined population. And this is on the risk. So what I'll talk about is in terms of risk difference. So that if you compare the proportion, the probability of having a successful outcome under treatment versus that probability under control, the null hypothesis is no better under treatment than under control. And you could ask the same question, pose the same null hypothesis for just a population 1. Now you could also add in for population 2 if you wanted to. But for us, we focused on just population 1 because there's better prior evidence that the treatment's going to work for these guys if it works for anyone. So population 1, which is small IVH population, and then the combined population of anyone with ICH. And so there are 3 design goals The first goal is to have at least 80% power to reject the combined population null hypothesis when the treatment effect is positive. So we said a 12.5% increase in the proportion of people who have a successful outcome. So, and that's on an absolute difference. So it's around 25%, 20 to 25% are having a successful outcome under current standard of care. We'd like to get that up at least 12.5%. So if you get that benefit in both populations, I want 80% power to detect that effect globally, to reject the null HTRC, 80% power. And I'd also like the design to have 80% power to reject null hypothesis for just subpopulation 1 if it's only that population that benefits. Oops. So those are the two goals. Now you can add goals, but that's also and you could apply a similar type of method. It'll get more complex. But this is maybe one of the simplest cases. And at the same time, I want the third goal is strong control of the family-wise type 1 error rate. And here I'm focusing on one-sided tests. So I'm using 0.025 as a cutoff, but you could also make it two-sided and 0.05. So those are the three goals. Seems like a relatively simple problem. If you ignore goal 2, then you can use a standard group sequential design. That would be probably a pretty good idea. But now that but if you put in goal 2, you want to make sure that if just subpopulation 1 benefits, that you have good power to detect that. That makes it trickier. Standard design focusing on just goals 1 and 3 will have pretty bad power for 2. Will not satisfy 2. And then the question is, what's the I want to minimize the expected sample size, and also keep the maximum sample size not too large either, and try to meet all these goals. And I'll compare. So next I'll give an adaptive design, and it's actually a pretty general type of adaptive design. And this is a new general class of adaptive designs. And we show in how to prove the type 1 error is bounded, or is the most 0.025. So we prove this for these designs. And that'll give you, and then we also have software that optimizes the design. Now it's not finding the true optimum. That's unknown how to do that. But we do optimize over a large subclass of designs where we consider different boundaries. We consider different sample sizes. But I'll make that clear in a minute. So first, I just want to give this general template for adaptive enrichment designs that incorporates group sequential testing. So you can have as many pre-planned interim analyses as you want, and you can space them however you'd like. And here's I'll tell you what happens at each interim analysis. So say you're at the third interim analysis, case 3. Here's what's going to happen. You're going to compute 3 statistics. So this all boils down to your decisions are essentially going to be based on 3 statistics at any time point. There's ZC. So the sub K that tells you the analysis you're on. And there are 3 statistics. There's ZC, that's the Z statistic for the combined population, and that's incorporating all the information up to that time point that you've collected. You also have Z1, which is just only using data for subpopulation 1, and you have Z2K, which is just for subpopulation 2. And in fact, this one and this one, you can use them to construct this one. But essentially, you have these 3 numbers. And the first step is you assess efficacy. So you do a hypothesis test for the combined population and for population 1. Using it, basically, there'll be a threshold. And I'll have a bunch of thresholds here, these U, little u and little l. The U's are the boundaries for rejecting null hypotheses. And then the L's are futility type boundaries. And those have to be all set beforehand. And that's what will be optimized later. So say that that's already set before the trial starts, and you're at analysis 3, for example, and say you've been enrolling the combined population up to then. And so the decision, so first you do this test. If the Z statistic for the combined population exceeds its corresponding threshold at that stage, then you reject that null hypothesis for combined population. And the same thing for population 1. If that statistic exceeds its threshold. Now, you don't need this one to reject in order to go into this one. They're separate tests. You could end up rejecting both, or just this one, or neither. If any of them are rejected, we'll stop. The trial will stop. Now, there are modifications to this. You could continue. There's a modified version where you're allowed to continue. Still, you control type 1 error, but for now, we would just say, as soon as you're able to reject one of these guys, the trial stops, and you're kind of that's it. So if that doesn't happen, nothing got rejected, then you assess futility for the whole trial. And there you look at the Z statistic for population 1. If it's below a certain futility threshold, then you stop the trial. And the reason the focus is on subpopulation 1 is that the prior information indicates that it's thought that if the treatment's going to work, it will very likely work for population 1. So if it doesn't work for population 1, that's enough to give up. Now a lot of these can be modified and still the general procedure will work, but it's tailored to this problem. So that you might decide to give up on the whole thing if the signal in population 1 is not very good. But if you don't, then there's there's not that much new going on in steps 1, 2, 3, but step 4 is maybe the more interesting, more exotic step, where you look at subpopulation 2, the Z statistic there, and if it's below its corresponding threshold, then you're going to give up on that subpopulation. If there's just not enough signal, and you're far enough along in the trial, there probably should be if there's an effect there, then you're going to give up on subpopulation 2. You're not going to enroll them anymore, and at each future analysis, you're just going to enroll subpopulation 1, and just do tests for subpopulation 1. You just check you're essentially shifting into a group sequential design with certain thresholds that I'll tell in a bit for population 1. So this is the case where you might give up on subpopulation 2 and just shift. Now you're just enrolling subpopulation 1. And I should say throughout this, I'm assuming that if you're enrolling the combined population, that you're enrolling proportional to the population proportion, the true population proportion. So in the case we have here where we're projecting it's a third of the population has small IVH and 2/3 has large IVH, I'm assuming when you're enrolling the combined population, about a third of your subjects have small IVH and about 2/3 have large IVH. And if you stop enrolling one of them, then you shift to just enroll one of them, then I'm not assuming you can increase the enrollment rate. I'm assuming, okay, fine, now the enrollment rate is going to go down, but you're just enrolling that one group. But when you're enrolling together, I'm not assuming you have this magical control over, I can double the recruitment rate of these guys while holding these fixed. I'm just assuming you're essentially sampling proportional to what's in the population. So these are the 4 steps, and then if none of these things kicked in, if you didn't reject any null hypotheses and you didn't stop for futility, didn't stop the whole thing, then you just go back to step 1 and keep going. You basically go to the next interim analysis until you've hit the final stage. And so any questions on the design? So my goal is to make it to use what's already there in group sequential designs, but now just add in, now you have another test. And so what has to be determined are, what are these cutoffs? What are the rejection thresholds for subpopulation 1, for the combined population? And what are these futility, these stopping thresholds? And we want to select them in a way that guarantees that those goals are met. And minimizes the expected sample size, and also doesn't make the maximum sample size very big either. And here I'll just show you what we did a pretty intensive optimization where I had at least 20 or 30 different parallel processors going on for multiple days to basically search over many types of designs, where we optimize the sample size, optimize these thresholds. There's so many of these thresholds, there's just too many possibilities to search over the whole thing. So I did make some simplifications. I essentially so here's the boundaries, efficacy boundaries for the combined population. So actually, let me just explain what this is. And basically, this sets the sample sizes up here in each stage, and down here we set all the thresholds. So I'll just tell the answer, but it's actually there's a huge amount of computation that went into this. But you can read in the paper, it tells all the details, and that's on my website. So here I took the case of 5 interim analyses. And so the first row is the cumulative sample size in subpopulation 1, then cumulative sample size subpopulation 2, and then cumulative combined population. And say so we can just focus for a second on the cumulative sample size in population 1. So here it's increasing at each stage. And if you were to immediately give up on population 2, this would just reduce to a trial where these are the numbers enrolled in population 1. And then the corresponding thresholds are down here, this H01 efficacy boundary. And this is proportional to O'Brien-Fleming boundaries. So I actually set every row here is, each one on its own is proportional to standard O'Brien-Fleming boundaries. The only thing is you have a proportionality constant that's different for each. And here you have the subpopulation 2 cumulative sample size. And so I'll just walk through very quickly what could happen in the trial. So say you're at the first stage. You enroll a third of the patients are from subpopulation 1. You have 93. You have twice that many in subpopulation 2 for a total of 280. And at the end of the first analysis, first you compute a z-statistic for the combined population and see does it exceed this O'Brien-Fleming boundary, but it's pretty high. So it has to be super strong, just like O'Brien-Fleming is kind of set to do, to reject. If it's above this, you would reject the combined population null hypothesis. You do a similar test for population 1. Is it above this threshold? Then you would reject that null hypothesis. And if either you reject, you'd stop the trial. And then you have these futility boundaries, which are here it's just 0. So this is the boundary to stop subpopulation 2 enrollment. So if subpopulation 2 z-statistic is negative or equal to 0, then you would stop that enrollment. And the same thing for population 1. If that thing is 0 or negative, you would stop the whole trial. And actually so that, I'll come back to those zeros. I mean, that's kind of aggressive in a sense. It's very, those thresholds are set to have a pretty good chance of stopping the trial if there's really no effect. And we optimized over these, and this is basically what came up as the best options. And now you notice once you hit stage 3, that's the last stage at which this particular design allows enrollment of subpopulation 2. So this design has built in that if it's the case that at the end of stage 3, you haven't been able to reject the null hypothesis, you haven't rejected anything yet. In particular, you haven't rejected for the combined population. You're just going to give up on population, give up for the combined population. So it basically sets this threshold to infinity. So it's forcing that if the trial has gone on to this point and you haven't stopped population 2, it's going to force that happens. And then you'll, if you haven't given up on the whole trial, you'll just continue testing for subpopulation 1. It essentially reverts to a group sequential design for population 1 with some, with higher thresholds than a standard to control the overall type 1 error. And the intuitive reason, so actually I tried in optimization, it includes procedures that don't have that feature built in, that just go all the way to the end. But this one it's much better. You do much better on expected sample size if you have a rule like this. Any guesses why that's the case? This is particular to this case where pi 1, the proportion of subpopulation 1 is a third. It's smaller than the subpopulation 2. I don't want to take a shot at it. It's tricky. This is just what the optimization algorithm produced. But there is some intuition for why that's the case. Garrett? Yeah. Yeah, that's fair.
So for 4 and 5, you're only going to be continuing to test the population 1?
That's right.
So you no longer include that, even if you didn't reject.
The
You no longer include the population, you no longer do it, even though you didn't reject.
Yep. So at the end of the trial, if you haven't rejected for the combined population at the end of stage 3, that's it. You give up on it. Well, that's right. So at the very end, if you enrolled to the very end, if there was no stopping at all that took place, then you would have 576 from subpopulation 1 and 560 from subpopulation 2. So yeah, it's about the same in each. And this particular design does basically, it's if you don't see the signal, if it's at this point when you stop enrolling subpopulation 2, that's the last chance to test the combined population null hypothesis. Now, you could consider a different design that still, even though you don't recruit people from subpopulation 2 here, you could still, you're getting information on subpopulation 1. That is getting more precise, your estimate for subpopulation 1. So that could help refine your estimate for the combined population. So you could incorporate a test of that. The only thing is that's going to increase the type 1 error. So you have to pay. There's going to be a price paid for that somewhere. So basically you could, and that's actually one of the directions for future research is to look at what happens when you continue testing that, which that could have good properties. But you just have to, there's going to be, basically it's going to up, some of these thresholds are going to have to go up to control type 1 error if you do that. So there's going to be a trade-off. So the question is, what is the trade-off? So that certainly could be true. And that is one of the next areas to look at is what happens in designs where either it's also possible to continue, even if you reject one of these, you can still continue. That will increase the expected sample size if you do that, but it will also increase your power, so that it's a trade-off. And it's a good idea to consider that type of design. So it's another possibility, for sure. Yeah? That would be the idea that if you were to go on beyond, now this doesn't have this built in, but if you were to do a different design and say at the end of stage 4, now you've collected the proportion of the total subjects in your trial, the proportions are off from the population proportions. So you'd have to do some kind of adjustment. You'd essentially get an estimate for each population separately, and then combine them in proportion to the true overall population proportion. So so you would do that. So that's an interesting question for next step to look at. So let me just conclude by saying that when you compare, so there are three scenarios. The first is where the treatment benefits both at 12.5%. Second is where the treatment benefits only population 1 at 12.5%, no benefit for population 2. The third is the null case of no benefit for anybody. And here I compute the expected sample size of the adaptive design that I showed compared to two types of standard designs. One is basically the same thing, except I just remove the adaptive step. So I did the same optimization, but now I just rip out this step. You're not allowed to reduce to just one population. And then the other comparator is two just separate designs. One is enrolling the combined population. One is enrolling only population 1. You just kind of two separate designs. So those are sort of, you should be beating those. And you actually beat them. The average sample size is substantially lower for the adaptive design. I'm averaging over these three cases with equal weights. It's 635 compared to 885 and 722. And also the maximum sample size is also smaller for the adaptive design. So you're better on both. Now since I'll just leave it as this is only in the case of the 1/3, 2/3 population split. As you vary that, you can compare and see which of these types of designs, optimized in each case, which one is doing better than the other. And it sort of switches. There's a certain point where one type is better than the other. The adaptive design is always better than all of them. But at some places, this is as you change pi 1 from very small to very large. This is the adaptive design expected sample size, and these are the other designs expected sample size. It kind of switches. So there's some interesting behavior which has some intuition behind it. But overall, you can get some benefit. And potentially, for example, using Gary's idea, you can get even more benefit for all the designs by adding something like, especially for the adaptive, by adding the feature he suggested. And then there's a software tool we have to allow you to plan this. But because of time, I'll stop here. But and I want to thank everyone on the PACES team and a few additional people at FDA, Jingyi Ku, Hope Lamkin, and Gary Gray, who gave great feedback, and also to thank Jody Siegel and Emily Little on the PACES team. But thank you very much. Sorry.
If you have a few minutes.
Yeah, I can stay around for anyone.
Individual questions up here.
But feel free to leave if you need to. I'm not offended by it.
I was kind of surprised at the end where you brought in the proportion of the population and how you had to know about that.
Oh, you don't have to know it in advance. So you can actually. So it's a really good question. It's a great question. So in all of this, we planned it with a proportion in mind. But it can be, basically you can generalize this so that you only set this row and you just let the population come in. And you could be wrong. That's a pretty big issue, I think. And it's often ignored. But in the paper, we address that you could be wrong, for sure. You're not going to get it exactly right. And we look at, what happens? We generalize the design to make it so that you just see what population proportions come in. So it could be you go to 280 total patients, but this could be a little off. And then the thresholds are set depending on what these proportions are. But it's a fixed algorithm that controls the type 1 error. And we show that at least plus or minus 5 or 10%, you still maintain the properties still hold up. You're always guaranteed type 1 error control. Still the power is holding up. But if you're way off, then the power could be hurt.
And is it problematic if that proportion changes over the course of the trial?
It's assuming it stays the same over the trial. Yeah. So that could be an issue if it changed. Yep.
Yeah, and in different situations, that's very likely to happen. As you know, that kind of people enroll the easiest patients first, and then they're beating the bushes to get patients.
Yeah, it's a good point. It's a good point. And actually, I think you can generalize all this to handle it being different, but then it gets a little tricky. What is the population of interest that you're testing then? So you have to, what are you saying up front, that what is the combined population null hypothesis? If the proportion is changing over time, what is it defined as? So that even defining what is the null hypothesis gets kind of tricky in that one. So, but yeah, that's harder. This doesn't make that assumption. And it does make another important assumption that I should say, that this is all based on being able to observe the outcome relatively soon after enrollment. If there's a long delay, the information you have at an interim analysis will be much less. And we do have results that will be in a paper that's coming out pretty soon with Hui-Chong Q on designs that handle delay, essentially using these ideas but extending to the case of delay. We show you can still get some benefits depending on how the duration of the enrollment period and how long the delay is. If it's extremely long delay, then you can't get much benefit. But there are cases we look at where you can. So, but it's a good, very good point. Jay?
Yeah, this isn't really a question, just something you might want to think about. For estimation at the end of a trial like this, I can see people saying, well, if you stop subpopulation too early, I can see people saying, well, the only estimation we need to do is in subpopulation 1. But should we throw out the subpopulation to people completely in estimating an effect size and a confidence interval? I would think the latter is a more robust estimate of effect size, especially since you were relying on the MRS to dichotomize and negative variation in that. And some people are in the wrong group because on that one day the measurement ended up on one side, it could have easily ended up on the other side. So it's more complicated. But then if you include subpopulation 2 in the estimation, would you weight it down in some way for optimization?
It's a tough question. So optimizing the analysis at the end is going to be pretty. That's a tough question. For estimates, we just use the maximum likelihood estimate for each when the thing stops for each population. But there is a paper that with Xing Tsai, one of the students I work with, where we construct simultaneous confidence intervals for population 1 effect and the combined population effect whenever the trial stops. So it's set up so that whenever the whole thing stops, we're going to give you point estimates and confidence intervals for both that have overall its correct coverage guaranteed. And we show that if you use a standard confidence intervals that ignore the fact that this is an adaptive design, you can have under coverage, definitely. And the increase is something like 15% is roughly how much you have to expand to guarantee correct coverage probability. The thing is that you have to expand even in a standard group sequential design, not that much, but some, to have that be true as well. Yeah, that's a really good point. How do you do the best estimation? That's all a tough problem. Nick?
What's the name of the software tool?
It's called InterAdapt, and we're going through the beta testing. But if you want a copy of it, you can email me. I'll send. If you want to test it out, that would be great, actually. And I should tell, it's Aaron Fisher and Harris Jaffe. I didn't get to talk about the software, but they created this amazing user interface where you can upload your own data set. It will extract some features and allow you to play around with different parameters and see how the different designs do. That's the other issue, is how, even though in particular cases you might show that one design is better than another, you really want it to be in a wide variety of settings. Because you're not going to be sure before the trial starts on a lot of things. So you'd like to show that it's really robust in the sense that one design is giving you advantages in certain cases, and that those hold up as you vary a bunch of the parameters. So that's some of the future work to do that. So for example, if the population is changing over time, then that's trickier. So
I'm just going to call it a halt because I have to get on a conference call, and I want a chance to say thank you personally. So thank you, Michael, and please come up if he has time and you have additional questions. Is that all right?
yeah, that would be great. And thank you all for the feedback. This is very useful, the feedback you've given.
That was great.
questions.